This is what a neuron cell looks like. The first part of the neuron structure we are going to look at is the dendrite. A dendrite is an extension of nerve cells that propagate electrochemical stimulation from other neurons. Next we have the axon. An axon is a long projection of nerve cell that conducts electrical impulses called action potentials away from nerve cell bodies to another nerve. Cell body pictured here is a soma. A soma is a spherical part of neuron that contains the nucleus and connects dendrites to the axon. This part of the neuron is called the myelin sheath. It is an insulating layer surrounding the axon that increases speed of conduction and is made of Schwann cells. The cells that make up the myelin sheath is a Schwann cell. These cells assist in nerve regeneration and increase the speed of propagation. Once you go down the neuron through the axon terminal, then you reach the synapse. In the synapse are synaptic vesicles. These vesicles store neurotransmitters that are released according to voltage-dependent calcium channels. This neuron is a presynaptic neuron, while this neuron is a postsynaptic neuron. Presynaptic neuron is the end of the neuron that fires neurotransmitters to another neuron's dendrite as a result of an action potential entering the axon terminal. Postsynaptic neuron is a neuron in which an electrical impulse is transmitted across the synaptic cleft from a presynaptic neuron. The electrical impulse is through neurotransmitters. This is the synaptic cleft or gap. It is the space between a dendrite and an axon terminal where chemical neurotransmitters transmit excitatory or inhibitory signals.
Saltatory conduction is a type of nerve impulse conduction in which action potentials travel down an axon. These impulses move across the nerve fiber by jumping from one node to the next. This jumping mechanism helps save energy by decreasing the use of sodium-potassium pumps in the axon membrane and use the increased speed to allow the organism to react and think quickly. Moving from one node to the next also increases the conduction velocity of the action potentials. Saltatory conduction requires both sodium and potassium channels to function.
The resting membrane potential is at negative 70 millivolts because there are more sodium ions outside this neuron and more potassium ions inside the neuron. Sodium-potassium pumps move 2 potassium ions into the cell and 3 sodium ions out of the cell to keep the charge inside the cell negative and help the resting membrane potential remain constant.
Once the signal has been passed down the axon by depolarization, the axon needs to be repolarized so that they can send a signal again. To do this, sodium ion channels close and potassium ion channels open so that potassium can flow out. Then, the sodium-potassium pumps take 2 potassium and 3 sodium and put them against their concentration gradient, which repolarizes the membrane. The refractory period is the period after the neuron fires, where the cell is in a resting phase and cannot respond to additional stimulation. In this time period, the neuron will not go through another action potential, no matter how strong the stimulus is.
Neurotransmitters are labeled as excitatory when they make the postsynaptic neuron more likely to fire an action potential. When these are released, they open ligand-gated sodium channels which allow sodium ions to enter the cell. The depolarization that occurs is called the excitatory postsynaptic potential. This results in the cell being more positive than the surroundings.